In this screencast, we are going to talk about cell signaling. So the gist, the main overview here is that cells actually talk to each other in a what's called chemical language. And the idea here is that there are little chemicals that are released by cells, or maybe they're in the environment, that can actually send messages to other cells. And so we call these little chemicals ligands. These are the signaling molecules, and they bind to a very specific receptor in the cell, and then that causes all manner of change to take place inside that cell. So in class, we watched this TED Talk about how bacteria can communicate with one another. It's called quorum sensing, and how they basically figure out, are there enough of us to actually take down a host? And it was a really interesting TED Talk and kind of a good overview of this topic if you're iffy on it. So, the different types, there's autocrine, where the cells release a chemical that then affects the original cell releasing it. There's juxtacrine, where the cells are in direct contact and can affect each other through direct contact. Paracrine is where they only affect nearby cells. So that quorum sensing that I talked about in bacteria is an example of that, where one bacterial cell can send messages that affect nearby individuals. And then endocrine is where molecules are released into the bloodstream. And then from the bloodstream, they can affect cells anywhere else in the body. And so in endocrine signaling, we call those chemical messengers hormones, which I'm guessing you've heard of. And that's what I want to focus on for the rest of this screencast. So the key point is that all of the hormones contact all of your body cells via the bloodstream. But only certain cells have the appropriate receptor to respond. So, for example, we've got hormone A, which is circular. We've got hormone B, which is triangular. This cell will only respond to hormone A because it has that nice receptor complementary in shape to that hormone. This cell over here will only respond to hormone B because its receptor is complementary to that cell, that shape. This cell will actually respond to both because it has receptors for both. So again, it's very specific. So it's kind of like Facebook. If you're not familiar with Facebook, think of Twitter for old people. You put a message out there, and then people either respond or ignore it. Another key point is that the same hormone can affect different types of cells in different ways. So, for example, if you add epinephrine to I keep covering up, so so that first one, I'm trying to get rid of that bar on the bottom, the liver cell. If you add epinephrine to a liver cell, then that will cause glycogen to break down. If you add it to a skeletal muscle, blood vessel, that middle one, then it causes the blood vessel to dilate. If you add it to an intestinal blood vessel, then it causes the blood vessel to constrict. So the same hormone can have different effects on different cells, which means that there must be other molecules involved between the hormone and whatever effect it ultimately causes. So there's two broad classes of hormones, steroid and water-soluble hormones. Steroid hormones affect DNA expression to initiate long-term changes in the cell. You've heard of some of these. Testosterone and estrogen are two steroid hormones that affect the human body. Notably, they cause the development of secondary sex characteristics during puberty. So these are pretty big, broad broad changes that happen as a result of these hormones. If you look at these, you see a lot of carbon-hydrogen bonds. So these are hydrophobic, meaning they can pass through the cell membrane. So they're lipid soluble. And then these bind intracellular receptors, that is, proteins inside the cell. And so here's what happens. The hormone is floating around the bloodstream, la, 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 la. And if it comes across a cell which has the appropriate receptor, it will interact with that receptor. That receptor will then travel into the nucleus, which again is no problem because the nucleus has these nuclear pores, these big holes, so it can pass right through there. And then it binds to DNA and acts as a transcription factor. Now, the transcription factor will then cause specific genes to be transcribed to make new protein products. Now, when you first learned about this, we hadn't talked about transcription. You didn't know what transcription was, so we had to kind of tiptoe around this a little bit. But now you do understand that hormone receptor complex acts as a transcription factor, and then you may recall from Unit 6 that helps RNA polymerase bind to the promoter so that it can transcribe those genes. So basically, testosterone or estrogen is produced in larger amounts during puberty, and then those hormones interact with the DNA and cause the transcription of the genes that trigger the development of secondary sex characteristics. And so that's why, in males we see a deeper voice, the beard, pubic hair. In females we see breast development, pubic hair, the onset of menstruation, all those things. New protein products are being produced as a result of these steroid hormone actions. The other class of hormone then are water-soluble hormones. These affect enzyme activity to initiate short-term changes in the target cell. So remember, steroid hormones affect DNA expression to initiate a long-term change. Water-soluble hormones affect enzyme activity to initiate a short-term change. So you've heard of many of these too. Epinephrine, which is basically adrenaline, insulin, maybe you've heard of melatonin. These are typically proteins, and so they may or may not be able to cross the membrane depending on their type, but if they're water soluble, then they won't. And then they they engage in what's called a signal transduction pathway, a series of steps whereby the binding of the signaling molecule results in a cellular response. So this is where things get complex. So an example that we're going to walk through in detail is epinephrine. This is basically adrenaline, triggers that fight or flight response. So if you're walking down the street and somebody leaps out and goes, "Blah!" and you freak out, your heart rate starts, jumps up, your eyes bug out. That freak out is called the fight or flight response. And one of the things associated with that response is that epinephrine ultimately causes glycogen, this storage carbohydrate, polysaccharide, to break down into glucose, which is a monosaccharide. So how does it do that? Well, you don't need to memorize these specifics, but just for illustrative purposes, we'll go through this as an example, just so you understand how complex these can be. So the signal transduction pathway for epinephrine, and again, this is specific to epinephrine now, has a number of players. There's a G protein that when activated, here's the G protein right here, when activated, it's bound to GTP, but when inactivated, it's bound to GDP. There's a G protein coupled receptor that when activated by a ligand is what actually activates that G protein. There's a protein kinase. So kinase is a term you should know. That is an enzyme that transfers phosphate groups from ATP to a protein. And kinases activate specific proteins in the cell. This is a common way to activate proteins. And again, by adding that phosphate group, you're essentially changing the shape of that protein in some way that activates it. And then there are second messengers. A second messenger is a small molecular ion that is kind of an intermediary between the activated receptor, here's our activated receptor, and then the activation of other proteins. These are typically very small molecules, and we want them to be small so that they can diffuse quickly throughout the cell. Smaller molecules travel faster throughout the cell, so they can carry out their effects more quickly and efficiently. So here's how it goes down. You look at this, a little part of you dies inside thinking, oh, good Lord, that's so complicated, but let's just take it little by little and we'll figure it out. So step one, what happens? That epinephrine binds to a G protein coupled receptor. That G protein coupled receptor then activates the G protein by allowing GTP to bind to it. So here's our activated G protein. The activated G protein then activates another enzyme. We don't care what the name is. It just activates another enzyme that converts ATP into cyclic AMP or CAMP. That is our second messenger. So far, just think of it like dominoes. One domino knocks over another, which knocks over another, which knocks over another. Like it's just, you don't need to worry about all the details. Just one thing triggers another, triggers another. Um, and then that causes a signal cascade to occur where CAMP is going to take this inactive protein kinase and activate it. Once active, that will activate another one. And once active, that will activate another one. And ultimately, that is going to activate our target enzyme, in this case, the enzyme that takes glycogen and breaks it down to glucose. So again, big, large, complex, scary diagram. But just think about like dominoes. One thing triggers another, triggers another, triggers another, triggers another. You don't need to memorize the specifics of any particular pathway. You just need to understand the general principles around them. Typically, on an AP test, they will give you an example in a diagram, and you have to interpret what's going on, or sort of understand these key concepts around that. So what are these key concepts around them? First of all, they always have these three main steps. There's reception, where that signal molecule binds to the receptor. There's transduction, where we relay that message to the interior of the cell. And then there's the response, which is the particular change in the cell. So in this example I just went through, reception would be epinephrine binding to the receptor. Transduction would be the activation of the G proteins, the cyclic AMP, the protein kinase cascade, and then the response would be glycogen breaking down to glucose. Okay. Another key point, amplification occurs. Note that each enzyme in the pathway activates many enzymes in the next step. So these numbers on the side indicate that. So basically, one binding of epinephrine stimulates many G proteins, which ultimately results in the production of 20 cyclic AMP. Those cyclic AMP activate cause the activation of 20 protein kinase A's, but then those protein kinase A's, remember, they're catalysts. So they're going to go nuts. They're going to keep doing this reaction over and over and over and over. So these 20 protein kinase A's result in 100 active phosphorylase kinases. And then those 100 mean 1,000 of these are activated. And then that means 10,000 of this reaction is actually happening. So the binding of one epinephrine molecule results in 10,000 reactions of glycogen being broken down into glucose. So this is how these responses happen so quickly. It's that amplification step. Another key point is that there's a lot of variation in these. And so, I won't go through all these right now, but just know that variation exists. You can look up this on your own. And on the AP test, typically they give you an example, a picture of this. So if you understand these main concepts of how the signaling molecule interacts with the cell and reception, how transduction takes place through, with amplification and second messengers and these other things, generally, if you have those concepts, then you can interpret the situations quite well. So that's it for signal transduction and cell signaling. Thanks.